Good afternoon, sir. May I start, sir? Hello.
Please.
Ask Prathamesh to start recording.
Started, sir.
Okay.
Okay. Good afternoon, all of you. I am Dr. Arti Kamble, Assistant Professor, SGM College, Karat. First of all, I would like to welcome all of you in the lecture series conducted by Microbiology Society of India. Please join me welcoming in Dr. Chittaranjan Deb, Senior Professor, Department of Botany and Director of IQAC, Nagaland University, a leading expert in botany who will be sharing insight on the exploring microbes. I will introduce him within a short period, within a short line. He has published eight patent, and out of that, four is granted. He got the International Scientist Award 2021. He is also got the dynamic. He, hello. He also got the Dynamic Professor Award in 2023. He also have a lead Lifetime Devotion Award by MBSI in 2024. And he also has a teaching experience. So let's start his lecture. Hello, sir. Can you
Yes.
start your lecture?
Yes.
Thank you, sir. Thank you.
Yeah. Thank you, Dr. Deshmukh, Arvind Deshmukh, the All India President of Microbiology Society India, Dr. Arati Kamble, the convener of MBSI. Thank you for inviting me to deliver a talk. Now, what I'll is the slide visible? Can you see the slide?
Yes, sir.
Is the slide visible? My slide, presentation slide.
Yes, sir.
Yeah.
It is visible all for all, sir. You can start, sir.
Okay, fine. What I'll be in next 40 minutes, what I'll try to share with our friends who are present in this webinar. What we've been doing in last 7-8 or so, as the title is exploring microbes. So we have been working for activities, trying to look into the microbes, both bacteria as well as fungi, especially growth promoting, who are residing in the Musa, especially wild Musa Rhizosphere. How those bacteria and fungi can be used for sustainable agriculture or green agriculture. Now, question comes, why Musa? Now, all of us we know Musa or banana is a staple third staple crop globally, and in India, in all parts of the country, banana is grown. Different cultivars are grown. Now, one of the major problem with banana cultivation is maybe in a 3 to 4 years, the banana plus at
Audio problem, sir.
by am I not audible?
Yes, sir, you are audible.
Hello?
Please go ahead,
It's
sir.
not audible, sir.
Yeah.
There was some range problem to others. It's a
You are
range
audible,
problem.
sir. You are audible. Go ahead.
Yeah. So maybe please your bandwidth. If the poor bandwidth, then that might be an issue. Shall I go ahead?
Sir. Yes, sir.
Yeah. So, one of the major problems associated with banana. So every 3 to 4 years, we need to change the stock, buy fresh stock of planting materials. But when we started a journey, sometimes in 2017, 18, a question was bothering to a scholar, started sort of coming, joining for PhD. I just projection. Can you give me the answer why the cultivated bananas are so prone to the different pathogens? There is virus, bacteria, fungi. But on the other hand, the wild bananas which are growing in the hilly terrain, they stand alone without any issue. After there is no infection. So what is the reason? Can you give me the answer? Then the journey started from there. Let's look into. You can see in this panel the side plants are growing so beautiful in the wild, while on the other side the cultivated plot how they get spoiled in note in a couple of years or maximum years time. So we try to look into the now 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 question comes to what the robustness of the sphere that push to 20.
Hello, sir. You are not visible and audible, sir. Hello. Hello, Dev, sir.
Hello. Is there any problem?
Yes, sir. I will call him, sir. Wait.
Okay, some network problem, I think.
Okay, sir. I will call him, sir.
Yeah, he will join. He will join again.
Okay, okay, sir.
You call him, he will join. So he will join within 2 minutes. I welcome you all. Uh, Chittaranjan Dev is a very well-known scientist, and he is from one corner of India. Generally, we say that neglected area, Seven Sisters. And just 2 months back, he got award from Microbiologist Society. So
Sir,
let us
he will
hope.
join again, sir.
He will join. There is a network problem in his area. His
Yes, sir.
area is a totally hilly area.
Sir.
So he will join. And on next Sunday, Arti, you arrange one more
Yes,
lecture.
sir. On next Sunday, Gauri Wagmari's lecture is there, sir.
Very good. And that is a very important topic, patent,
Hi. Yes, sir.
because she is a master in patent.
Sir.
Yes, yes. And that
He
is
will
the
guide for patents to others, sir. Too.
correct. So many research workers should join.
In sir, in different areas.
Yeah. I will make an appeal.
Yes, sir.
Bhavesh Narkede from which place? Bhavesh Narkede. Dr. Bhupesh.
Yes, sir. Yes, sir. Good afternoon, sir.
Good afternoon. You are from which place?
Sir, Nagaland, sir. Nagaland. Nagaland University, sir. Lumami,
Okay.
sir. Nagaland. Yes, sir.
Very good.
Yes.
Chittaranjan Das has joined.
Yes, sir.
Very good.
Yes.
Chittaranjan Dev,
Yeah.
and please go on. And if
Yeah,
there is a
host
problem,
is not allowing to share.
yes, 1 minute. I will allow.
Yeah.
Uh, 1 minute. Arti,
Yes, sir.
you call Prathamesh because
Ah, yes,
he
sir.
can
Okay.
allow. I cannot allow. In the meantime, you start, sir. He will allow you. Don't worry. You proceed without PPT.
तुम्ही आला तर जाणार नाही. केदारच्या गाडी म्हणून जाणार. तुम्ही आला तर जाणार ना? आणि आपण आपल्या
Yes, yes, yes. बरं झालं भेट झाली. Just I'm making you sir host. I will allow you. Don't worry. Sir, I will allow you.
Can you allow me, please, to
Yes,
start?
yes, yes, yes. I will allow you.
Yeah.
Now you are allowed, sir. You can share.
Yeah. So, sorry, because of the weather condition now, bandwidth is very poor. So, we try to look in is the slide seen? Can you see the slide?
Yes, sir.
Yeah.
Yes, sir. Yes,
So,
sir.
the serial. So, what we have done the third generation pure culture of both bacteria and fungi, and then we have proceeded for the next level. Then these are some of the isolates of third generation pure culture. What we have done the molecular characterization of bacteria. Then these are the fungi. Then now I'll just here I'll share three, in fact, four different experiments of what we have done in last 4 or 5 years. Now this is one of the story with the wild banana Musa iterans. From there we have isolated over 60 different bacteria, and then we have based on the preliminary study we have isolated 25 bacterial isolates and then taken them for certain biochemical test and plant growth promoting test like phosphate solubilization, C04 production, IA production, then heavy metal tolerance, ammonia production, salt tolerance, etc. And from after all these studies, then from these 25 species or isolates rather, we have prioritized three isolates: EZ11, EZ27, EZ30. These three isolates, and then we have used 16S rRNA sequence for molecular characterization, and they are revealed that as Pseudomonas putida, Coccinia aerogenes, and Pseudomonas montili. And then these three isolates were taken for checking for growth promotion traits in a model plant scissor. Now, as this, you can see here the different parameters, including the gene bank accession numbers of these three isolates, and then below panel you can see the different some of the tests what we have done. There are many other, but here I am just for this discussion. I have added just few of those tests like catalase, citrate utilization, migrate test, starch hydrolysis, then different sugar fermentations like sucrose, maltose, dextrose, glucose, mannitol, and so on. Then you can see the different qualitative and quantitative analysis of growth promoting traits like PSI, phosphorus index, phosphorylation quantitative studies, IA production, C04 production, ammonia production. And in here you can see out of these three isolates, only Pseudomonas montili was able to produce IAA, while other three bacteria or isolates are able to both produce C04 as well as ammonia. Then these are some of the photos of phosphate solubilization in the upper panel, then ammonia production, and then C04 production. These are some of the routine tests we have done it before we go for in the model plant. Then it's a salt tolerance. Now, interestingly, you can see we have gone from 2% to 14%. Now, one of the isolates, your EZ11, you can tolerate, surprisingly, it could tolerate up to 14% sodium chloride, such a high level of sodium chloride tolerance these bacteria exhibited. Even the others has shown up to 6% to 10% easy there, but isolates EZ11 could tolerate up to 14% of sodium chloride tolerance. Then we have done at least 7 different heavy metals tests. But here I am showing the data for only cadmium. And now again, EZ30, you can see it can tolerate up to 280 microgram per ml. Again, it's a very high dose of cadmium these bacteria could isolate. Then after doing the preliminary study, what we have done, we have taken it to the host plant. Here in this panel, there are 4. The last G, you can see, is a control, and then you can see with the other 3 species, how you can see how the growth is promoted. Now here I am. In the next slide, I'll be showing how the vegetative growth is promoted. Then we have the even the production data, which is a part of the patent. And then here, what I'm showing is only the single isolate, but then we have done the consortia also, and crisscross consortia for all the 3 isolates. Now here you can see how against control, how these 3 different isolates, they impacted the growth of different growth parameters, how impacted these 3 different. So from here, not fine. Plus, we have shortlisted to plus. From there, we have again refined to 3 isolates, having a very low high tolerance level for sodium hydrochloride, table salt. Then these impacted positively under heavy metal production, the growth of growth as well as the productivity of the host plant. Then the consortia, which is part of the pattern, so I am not telling details of these 3 isolates, improved significantly the growth and the productivity under environmentally challenged conditions. So these are the part of these three below panel pattern, the three, one Indian pattern, two South African pattern, all are granted now. And then one other paper, the basis of this was a part of these publications. Then another story, we try to look into the coal mine area. In different parts of the country, we have large number of coal mining. Nagaland. In the Nagaland coal mine area, what we have done, we try to isolate from banana, wild banana rhizosphere. That's here in the study, what we have done, we have shortlisted 26 isolates on priority of from our 50 isolates. And then from there, we have done the preliminary exercises, study again to further scale down the isolates for production, phosphate solubilization. And we have shortlisted the 7 TSU 1, 11, 12, 3, 4, 7, and 8. And then these are subject to molecular characterization consistence rRNA. Now you will see in this case, isolate could tolerate up to 8,070 microgram of heavy metal. Surprisingly, is huge can tolerate. So virtually this bacteria can tolerate any kind of heavy metal tolerant polluted area. And again, some of the isolate TS8 and TSU4, they could tolerate up to 12% sodium chloride. Now you can see then these isolates were tested for in two different host plants. In this experiment we have done in two different host plants. One there is the anode, and in this case we have done it without heavy metal as well as heavy metal. In this case, what data I am showing is with cadmium and copper and introduced. Then you can see here, these are molecular character gene bank action numbers of these 7 isolates. You can see the pseudophore product by these 7 isolates. Then ammonia production by 7 isolates. Then pseudophore production by these isolates, all the then you have the sodium chloride tolerance level of 7 isolates. Now here, at a glance, you can see here the different, some of the bonding states like phosphate solubilization, ammonia production, sodium level tolerance level, as you can see, TSU4 and TSU8 tolerate 12% chloride, and here we have tested as many as 1 to 7 different heavy metals, some from heavy to moderate heavy metals like all, including you can see the tolerance level of different isolate highlights are been given, and here you can see how tolerant these isolates are. Now again you can tell panel help us and the different isolates here. C stands for habitat. T stands for only growth promoting bacteria. T stands for both the bacteria as well as habitat treatment. So you can see in both the brassica as well as less flower control for the other groups is noted. So again, you can see how here I am showing for both the species the shoot promoting traits of by different isolates. So again, here the lead species what we have found bacterial isolates is Bacillus cereus, Bacillus promulgatus, Latiota, then Pseudomonas carinus, Cilia, very cool. International first ever report from Northeast India. There was no previous report about this bacteria, heavy metal tolerance, salt tolerance. So these are first ever report. So we have isolated these from coal mine polluted area. So again, these injections of good right can improve significantly both the vegetative growth as well as the productivity of both the host model plant, Brassica and Helianthus annuus in heavy metal area. So this what from here the takeaway point is these isolates can be well used for bioremuneration in the contaminated soil or coal mine polluted area with animals or contaminated. Then third case, what I can here number of cases here is a case with another difference is Alvicina. Here in this case, what we have used, we have different host plant for model plant for Phaeolus vulgaris. So we have kept on changing the host plant. So here also we have isolated 20 different PGPR, and then they've been tested for different growth traits like phosphate solution, IA production, siderophore production, salt and heat as explained discussed in the past two cases. Then all these from there we have prioritized here the different ISO. You can see RZ27, RZ23, and RZ5 and RZ3, and they have been identified as Barcolderia cepaciana. Again, the first report from Northeast India, then Pseudomonas Pseudomonas orientalis, and then these tested and you can next one you can see species wise they are they can solubilize phosphate like all the four different species given here like acids Pseudomonas orientalis up to 222.1 microgram followed by Bacillus capacian 222 microgram, then Microbacterium is 71.5 centimeters poorer, then Pseudomonas taiwanensis 19.0 microgram per. So these four isolates again you can see here again RZ27 again can tolerate up to 14% sodium chloride followed by RZ5, RZ23 each having 2% sodium chloride. RZ3 6% sodium still is a very high concentration. They had a significant siderophore production ability, or so which can indicate the iron mobilization. This RZ23 33.34, followed by RZ23 29.07, then RZ5 27.20. So again, in this case, you can see the Agrobacterium lerimoria and Pseudomonas orientalis could tolerate chromium up to 1840 microgram per ml. Again, it's a very, very high dose concentration, followed by Bacillus 1810, still very high. Then Pseudomonas taiwanensis 1300 microgram per ml. Then we have used these all three bacteria in different combinations as a crisscross for consortia, and then we have a very promising result. Then we have used it in Phaeolus vulgaris as a model plant. Then of these four isolates, the isolate Z RZ3, that is Pseudomonas orientalis, significantly enhanced shoots and root length as well as the biomass and also the productivity. So I am not presenting here the productivity because it has gone is a part of the patent which is already under examination. So these again show as before these results suggest that these isolates could be used successfully, potentially as bioinoculant to improve the plant growth and development. So again, here you can see the salt tolerant level of different isolates. Then phosphate solubilization, sulfur production, ammonia production, and you can see seize control, and against this control, all these four isolates how they are promoted growth of plant. Seize control RZ5, RZ27, RZ3, RZ3. Then here you can see, so these are the accession numbers. Uh, uh, so the next, we try to look into bacteria. Then in one case I am showing discussion is the growth promoting fungi or PGPF. Make the so we have 60 plus isolates. From there we have considered 57 third generation, and then from there we have taken the top 9 top performing isolates, and then they're based on 18S RNA and ITS sequences. They've been confirmed here. The data what I am projecting in this slide is based on 18S RNA sequencing as identified trichotoma atroviridic, trichotoma viridi, and all. So these are all the 9 isolates we have taken into consideration. Then in this case, we have used for different host plant we have taken. One is taken for consideration. So here the data was a photos presented here is for beta vulgaris. Then inside the vulgaris, how you can see against control how they have affected the growth and development of the 9 isolates. So the vulgaris and vulgaris, how they impacted the growth of the different parameters of basically parts of plants, both the whole plants like root length, shoot length, fresh weight and root dry weight as against the green are shown as a control against control compare the growth. Now here again we find we have done we have in calendar 15 species, 12 are on top. So we work with all the 12 species. Now, as of now, we have completed the work with only 5 species. And these species has given large volumes of bacteria as well as fungi as potential to be used as from microbes, whether it is for normal evolution or for environmentally challenged condition like heavy metal tolerant condition, well polluted. So this findings reveal that the wild Musa rhizospheres are storehouse of large number of bacteria and fungi tolerant to different heavy metals, uh, sodium chlorides or other salts having plant growth potentials. Now, so these both bacteria and fungi can be used therefore as biostimulant or bioinoculant for sustainable agriculture, because these days we are talking about to reduce the fertilizer, chemical fertilizers. So we can use as biofertilizer or biostimulant or bioinoculant so that we can get rid of or the harsh chemical fertilizers so that we can move towards green agriculture or sustainable agriculture. Now again question is we have out of 15 we have explored only 5, so remaining 10 species, the one we complete the episode, it will tell us more, and also what we feel we need to explore these microbes in different altitudes and different agroclimatic environments. I'm sure we'll be getting many more new isolates might not be known to the science till date, so we'll be able to do it. So let us. So what I would request, many of us might be working on sustainable agriculture. Then PGPR is a trend now going on. So not only wild banana, these other wild species can be considered to mine the growth promoting microbes for green agriculture. So with this, I'll stop here. Now I am open, and I'll thank Dr. Arvind Deshmukh and his team to give a chance to speak, share our one of our research activities in the university. Thank you.
Now the session is open for the discussion. If you have any queries, please ask him.
Yes. At the outset, I'm thanks to Chittaranjan sir, and today's lecture is recorded. We are circulating it within 2 days. Okay. So
Yes.
if anyone want to ask the question,
Good
he or
evening, sir.
she, yes, yes.
Yeah.
Sir, I'm from Punjab Agriculture University, Ludhiana. Sir, I want to know
ma'am.
whether you prepared a solid carrier-based biofertilizer or a liquid biofertilizer using these isolates, sir.
Ma'am, we have filed 7 patents, out of which 4 are already granted. Now on, we are working both on liquid carrier as well as semi-solid and even a dry carrier. Now, you can we can communicate some part, and I'll share with you the details. We are working with on low-cost carrier, so that as of now we got the success. We can take this carrier, keep them, this bacteria as well as the fungi, at least for 4 to 5 months, but we wish to improve the lifespan of this carrier, where otherwise what is many people what we are doing on work on biofertilizer. Recently we are discussing the same point at Balsar, with where Dr. Deshmukh also was there in one of the conference. The main issue we are talking about is failure. What we are giving, scientists are giving to the farmer. Farmers are failing in getting the things result. So where we need to work on the carrier, as rightly pointed out, we are working on liquid also, semi-solid also. But I feel the hint what we have got success is dry and low cost carrier would be better. But I wish I can take it to at least 6 months longevity, so that farmers can easily with ease. So rest I can discuss with you. I've given my email ID, or you can anytime you can give me. Anyone wants to note down, you can note down my point and number
Hanji.
also.
Yes, sir. Please share your email ID, sir.
Yeah, DEB.
Yes, sir.
I'll share my first slide, then it will you'll get it. Okay.
Right, sir.
I'm
Sir,
sharing
and
my
your number, sir?
first slide. Yeah, now here you can see the slide.
No, sir.
No, sir, it's not visible.
Okay, DEB.
Now it is available.
Yeah. You can anyone who is can note down the email ID. We can anytime we can discuss it for academic purpose. And mobile, if anyone to wish to note down, please note down.
Yes, sir.
I'm typing it here. 87875 Can you see?
Yes, sir.
Below the title, 8787576859.
8787576885. Many thanks for your talk, sir. Many thanks.
6859.
Good luck, sir.
Yeah.
Sir,
So anyone
thank you very
interested,
much.
always we can discuss.
Yeah, sir, Dr. Sunil here from Punjab, from Chandigarh, sir. I would like to ask a question, because population is very big in India, right? So is it there?
Yeah.
Is it this your particular fertilizer is increasing the production? Is there anything you have seen that the productivity is more with this fertilizer, this
We have
thing?
filed the patent, as I says, we have seen 2.5 times under agricultural challenge condition, productivity enhances.
Okay, so is it
We have,
means it is
we have filed 8
increasing the
patents.
production?
Huge, significantly 2.5 times under
Okay.
agriculture, agroclimatic challenge condition, even heavy metal under experimental condition. Now we are taking it a medium scale experiment, taking it to the Zoom field. Now one scholar is working on sustainable Zoom cultivation. So there we are taking our outcome, even the patents what we grant, we are taking it to the Zoom field, how we can use these protocol what we have developed on the isolates and the consortia taking it to the Zoom field, if we can make it Zoom more sustainable.
Yes, sir. Very good. Very nice work, sir. It's very valuable for coming future production and for farmers.
Yes,
It will be very
yeah.
farmer
Even
friendly
anyone
and
want to collaborate, we welcome collaboration also.
yes,
We'll be
sir.
happy to work together. Yeah,
Okay, thank you, sir.
yeah. Any more?
Sir, there is a one question in chat box, sir.
Chat box.
It's a question from Shivangi.
Yeah. Can we use here some response surface methodology for optimization of bionuclear? If yes, how we can? Yes, we can use response surface methodology, but we have not tested it yet. But definitely we can take it forward. Shivangi, Javid. Question is from Shivangi, right?
Yes, sir.
Yeah, we can discuss more on it. We can definitely we just started. We have not completed the work. We are working on it. So you can, as a number, you have given email ID. You can we can discuss on it. You can communicate. We can even I can join my research scholar, Dr. Moon Tatung, who is doing her postdoc on this topic. Any more?
Anyone asks the question, please ask him. If there is no question, sir, can I conclude the session?
Yes.
Deshmukh, sir? Hello? Sir is not there. Okay. Thank you, sir. We sincerely appreciate Dr. Chittaranjan Dev for sharing their valuable insight with us about the microbes from the world Rhizosphere Musa for the sustainable agriculture. And you are also explained the siderophore production, plant growth promoter. It's a very knowledgeable things. Thank you, sir. Thank you for enlightening and provided your thoughts and your knowledge with us. Sir, though there is a problem in between, but in the your thirst for knowledge, there is a no interruption. So I'm also thank you again, sir. I'm also thankful, Professor Dr. A.M. Deshmukh, sir, and Microbiology Society of India for giving me this an opportunity. Thank you, sir. Thank you all of you for joining the lecture. And here I am declare that the session is over. Thank you. Thank you all of you. Thank you once again.